So the first talk today is by Bradley Balaton, a PhD student in Carolyn Brown's lab. Bradley is a native of Saskatchewan. He's currently doing his PhD in the Department of Medical Genetics at the University of BC. He also mentioned that he will be graduating soon, and he will be looking for postdoctoral positions, hopefully in the field of epigenetics and epigenetics reprogramming soon. Okay, so he's going to be on the job market soon, so keep your eyes out for him. And today we'll talk about X-chromosome inactivation across different species. Okay, so Bradley, here we go.
As Marco said, my project is an epigenetic investigation of X-chromosome inactivation across species. Okay, there. So X-chromosome inactivation is the epigenetic inactivation of one of the two X chromosomes in eutherian mammals. And this is to dosage compensate with males who have only one X chromosome. And so the X and the Y are both evolved from the same ancestral autosomes, but over time, the Y chromosome has lost many of these genes, and the males and females still want the same dosage of many of the genes that are on the X. And so early on in development, females are inactivating one of the two X chromosomes, and this is randomly decided which X chromosome to inactivate in each cell early in development. And so if you take a random sampling of cells, you'll get a 50-50 mix of which X is inactivated. Not all the genes on the X are subject to X inactivation. In humans, around 10% of genes are escaping X inactivation, with at least 10% of the expression from the inactive X compared to the active X. And there's another 10% of genes which are variably escaping X inactivation. And so they differ in their X inactivation status across tissues, populations, individuals, or studies. And in mouse, this number is much lower, around 3%, 3 to 5% are escaping, and maybe another 4% are variably escaping X inactivation. And not that much work has been done in other mammals. So looking at epigenetic marks, the ones highlighted here are the ones that I will be talking about. And in general, the marks which are associated with silencing are also associated with gene subject to X inactivation, and marks associated with expression are associated with genes escaping X inactivation. And so here is a figure of the inactive X to active X expression ratio on the X-axis and DNA methylation on the Y-axis. And you can see a nice anti-correlation where you either have inactive X expression and low DNA methylation, or moderate DNA methylation and no inactive X expression. And this is similar across human and mouse. And we used these data sets to establish DNA methylation thresholds so that we could use these to call X activation status in other species. The problem here, as I stated before, with using the inactive X expression is that if you take just an average sample of cells, you're going to get a 50-50 mix, and then you can't tell which X is the inactive X and which X is the active X. And so in humans, we're using cancer cell lines for this. And for mice, you can do XIST knockouts so that they always have the exact same inactive X. So this is DNA methylation across species. I have female DNA methylation on the X-axis and male DNA methylation on the Y-axis. And you can see generally similar patterns across species. So you have your genes which are escaping X activation with low DNA methylation in both sexes, and then genes subject to X activation that have moderate DNA methylation in females and low DNA methylation in males. And then you have your genes that are just silent and have high DNA methylation in both. And so you notice here we have whole genome bisulfite sequencing with one reduced representation bisulfite sequencing data set. And then we also have a number of primates that have been run, plus dog, that have been run on the human 450K DNA methylation array. And these have been filtered to only include probes that map back across between species. And I'm mining other people's data. This is not generated by me. So this is the X inactivation status calls that we get with our data sets. You can see here that whole genome bisulfite is in general a lot better at making these calls than the 450K array. And that's probably just due to the amount of filtering we have to do, and also just because not all genes have a probe on the array. So we get around 340 X inactivation status calls per species on average. And so most of these species, if you look at the number of calls per X inactivation status, have around 10 to 15% of genes escaping. DNA methylation, we don't usually find that much variable escape from X inactivation. Only horse here has 10%, when we find in humans 10%, but I guess that human number is also including differences across studies, which this is only one study. And then looking at gene subject X inactivation, things in general, or the species in general, have 80 to 90% of gene subject X inactivation, except for mouse has more, and horse here, because it has variable escape, has less. And then we wanted to next look at genes which are discordant across species. And a couple things to point out here. So these green rectangles show that these genes are adjacent to each other. And so you notice that a lot of these genes which are discordant across species are in little clusters of genes that are adjacent to each other. And another thing to notice is that within these clusters, these genes also tend to have similar X inactivation statuses with each other across species, and that a lot of these genes here tend to escape in primates, and a lot of them are also primate specific, so they only escape in primates. And then you also have one gene here, KDM5C, which we've noticed is subject X inactivation in this group here, cow, sheep, goat, and pig, which are all from the same family or order or something. So next we wanted to look at the features which are enriched with different X inactivation statuses. So line repetitive elements are often stated as helping to spread X inactivation, and so they're enriched, or often seen enriched in humans and mouse when examined. And so we see that in a number of species. And then DNA repeats are also enriched in genes escaping X activation in a couple species here, and LTRs are enriched at genes escaping X inactivation. CTCF was another feature that we found enriched at many genes escaping X activation, or in many species, and has been seen previously to be enriched at humans. Here is looking deeper at humans. We have the epigenetic marks that are looked at in IHEC. And so looking at the genes which we found variable escaping in our inactive X to active X expression analysis, we found 8 variably escaping genes, and highlighted here are the ones for each mark that have been found significantly different between the samples escaping versus subject to X inactivation. And you can see some of these, it's quite an obvious change from the samples escaping to the samples subject to X inactivation, whereas other it's quite subtle. And then you can also see the distribution of the genes, which are the samples that we don't have the X inactivation status for, because they didn't have a informative polymorphism. And so some of these do look like they're on opposite ends of this uninformative range, whereas some of them you think that there's a large range of what's going on that's not captured by our informative genes. And so we wanted to try and include multiple marks at the same time to see if that captures the differences in X inactivation status. And so we decided to make a random forest predictor using all 6 of the epigenetic marks available in IHEC and with DNA methylation. And so from these, we could get around 75% accuracy for genes escaping X inactivation and around 90% for genes subject to X inactivation. And this was done on the samples generated by the Center for Epigenome Mapping Technologies. And we liked that one because it has all of the data in the same individuals and not just the same tissue. And then looking at the predictive abilities of the various marks or the contribution to the model. DNA methylation in females was always, or almost always, the top mark, with H3K27 trimethyl and H3K4 trimethyl in females also being commonly important. So in conclusion, we found X inactivation status often conserved across species, with mice being an outlier with fewer genes escaping X inactivation. Of the 15 genes found discordant across species, 5 of these were primate specific, with a number of other ones also escaping in primates. Genes that change the X inactivation status across species are frequently clustered. Many of the features found correlated with X inactivation status are consistent across species. Variable escaping genes did not have consistent epigenetic differences in the single genes, but incorporating multiple marks together, we were able to predict a gene's X inactivation status the majority of the time. And I'd like to thank everyone in the Brown Lab and on my committee and in our local bioinformatics office. Thanks.
Thank you very much, Brady. That was a very exciting talk. I have a couple of questions. I have a question about the transposons. Okay, I think you mentioned them briefly in one of your slides. And one thing that I was curious about was, do you think that what you're observing at the level of transposons is kind of causative of the escaping from X inactivation for regular genes, or is it just kind of a marker, basically, like things in the region is just kind of escaping because
yes.
Is lower?
So I think there's some debate. So the X chromosome is enriched with these line elements, and so I think that they're also enriched probably on the older parts of the X chromosome that are more tending to be subject to X inactivation.
Mm-hmm.
But at the same time, there has been proposals that because these marks are being silenced and are already recruiting silencing factors, that they do help the spread of inactivation, just because early in development, these the cells are already trying to silence these marks.
Okay, perfect. I have a question from Serge McGraw. He's saying, thank you, Bradley, nice talk. I might have missed it, but were similar cell types used for methylation data produced for species comparison?
So unfortunately, the similar cell types were not always found. Basically, I just combed through Geo and took every data set I could get per species. Mm-hmm. And so I believe all of the primates here are bone, there's like liver, all the cow stuff is agriculture related, so it's like mammary or meat. So yeah, it really depends on the species. If I was doing this myself, it would be consistent, but. Yeah,
for sure. Awesome. And then we have another question from Michael Johnston. It's the same question I was going to ask you as well, by the way. So you said that the genes that escape X inactivation are clustered together. Do they cluster within TADs? So if you look at if you overlay high C data, do they look like they're actually part of a specific TAD or a specific domain or compartment?
Yeah. So in humans, it's been done, and I think mouse too. So in general, they cluster within TADs. You do see a little bit where what is it? Some of the genes on the edges of the TADs might be different from the rest of the TAD, but it does kind of look like they go with TADs.
They go with TADs. That's awesome. We have a question from Jim Davey. He also said, great talk. So in human, ACE2, and many mu-related genes escape X inactivation. Does this vary from tissue to tissue and among individuals to the extent that these genes escape X inactivation in females?
So if they're wondering whether the ACE2 gene is variable, sorry, was that?
I think maybe there is a typo, Jim. If you want to send a different version of the question, I think so. It says, so I think what he's asking is if the extent of genes escaping X inactivation changes between tissues and among individuals.
Yeah, so there are variably escaping genes. So they are, there's a number of different studies. And so some of them see the genes change their X inactivation status across tissues. And then we do see some that looks like it's genetic and is different between populations. And I think ACE2 might have been in one of these discordant regions. I think it's near these genes.
Interesting, very interesting. And I
Oh,
think we
and
have
another thing related to that is that there has been some studies that show there might be some reactivation in immune cells, which would be relevant to that too.
Makes sense. Awesome. So I think we have time for one more question. And it's from Michael Johnson again. Is asking if the genes that escape X inactivation, if they tend to be very highly expressed, or if you see their expression or the transcriptional levels really to be spread across, a range of low, medium, high.
I don't remember seeing, like, I'm sure I've looked, and I don't remember seeing a clear correlation with escape genes being higher expressed. But with the methods we do use to look at genes escaping X inactivation, there is a certain threshold needed in order to be actually seen. So it does need at least some level of good expression.
Okay, awesome, awesome. Thank you very much for your talk and for the great answers as well. So now we're gonna move on to the PI talk. So it's really my pleasure to introduce Carolyn Brown, professor at the University of British Columbia. Carolyn Brown obtained her PhD in medical genetics at the University of Toronto. During her PhD and postdoctoral work with Dr. Willard at Stanford at Case Western Reserve University, she studied the processes of X-chromosome inactivation. And she actually really identified some of the genes responsible for this, including XIST. She was really a pioneer in the X inactivation field, okay? She's been at the University of British Columbia since 1994, and her group is focusing on the mechanisms of chromosome inactivation. Particularly, they're focused on studying the role of XIST RNA in initiating the heterochromatic changes that accompany X chromosome silencing, as well as DNA elements that allow some genes to escape inactivation. Okay, so our talk today is going to be about the functional domains of XIST for silencing by RNA. Okay, so welcome, Carolyn. I'm really looking forward to your talk.
Okay, thank you very much, Marco, for the introduction, and to Brad for introducing X inactivation. The Brown Lab is part of the molecular epigenetics group at UBC, and we're located on the 5th floor of the Life Sciences Institute. And as mentioned, we're interested in understanding epigenetic silencing, and X inactivation is a classic example of this silencing. But my talk is going to focus on the role of the long non-coding RNA XIST. So let's jump into XIST. The gene maps to the single region of the X chromosome that is required in cis for inactivation to occur. And it is expressed from the inactive X, not the active X, resulting in its name, and meaning that in karyotypically normal individuals, XIST is only expressed in female somatic tissues. We can see its localization pattern in this panel of images at the top from the Lawrence lab. So DAPI staining will often show the DAPI dense bar body. But with X paint, we can see that there are 2 X chromosomes in this female cell, but under non-denaturing conditions, XIST probes hybridize to the RNA, which co-localizes with the inactive X. So this nuclear location is quite specific, and in fact, the bar body, the inactive X, and thus XIST, is often found at the nuclear periphery or the nucleolar periphery. Now, as a long non-coding RNA, we expect the gene to be long and non-coding. And indeed, the longest open reading frame in the 17 to 19 kilobase human XIST is under 400 base pairs, and that is not conserved within the mouse, which is a 15 to 17 kilobase RNA. The lack of conservation of an open reading frame is not surprising, given that overall there's only about 70% conservation between human and mice. But they do share somewhat similar structures. So the first exon is over 10 kilobases in length, and the first and sixth exon contain a series of tandem repeats, which differ somewhat between species. They share a similar internal splicing pattern because these exons are derived from the ancestral protein coding gene. So the A repeat has been shown to be critical for the silencing function of the long non-coding RNA, and it's extremely well conserved across all Eutheria. There are 9 copies of that repeat in humans. The F repeat region is also an alternative promoter or enhancer for the gene, and is where CTCF and YY1 bind on the DNA. B repeats are actually microsatellites, and different microsatellites which are split apart in humans, but only one class in mice. The C repeat is actually, there we go, not a repeat in humans, but has expanded to be a repeat in mice and rats. The D repeat then has different monomer structures, and this structure is expanded in humans. The E repeat is somewhat larger in mice, and both humans and mice have this alternative short isoform, which takes out a fair block of the XIST 6 exon. And the short isoform has been shown to be functional in mouse, and we will also show it's functional in humans. Most of the work analyzing how XIST recruits chromatin modifiers and silencers to the inactive X chromosome has been done in mice. And this is predominantly because X inactivation occurs at the time of differentiation, and is therefore very difficult to study in humans. In mice, you can access those tissues in vivo, but importantly, in vitro, female mouse embryonic stem cells have 2 active X chromosomes, and upon differentiation, one of those X's inactivates. On the other hand, human embryonic stem cells generally retain an active and an inactive X chromosome, as they're more similar to epiblast cells. And then upon culture, that inactive X chromosome often erodes rather than reactivates. So in order to study X inactivation in humans, we have chosen to study the role of XIST in somatic cells. And this was a model system that Jen Chow established when she was in the lab, using an XIST cDNA from the short isoform and making it DOX-inducible, and then integrating it into FRT sites in the HT1080 male fibrosarcoma cell line. And what she saw was that upon DOX induction, if the XIST cDNA was integrated adjacent to a reporter construct, that DOX-induced XIST would silence, but in a reversible fashion. We can also see this effect again by in situ hybridization. So DOX induction of the XIST RNA leads to recruitment of chromatin marks. In this example shown is H3K27 trimethylation. So in order to understand this silencing, Jakob Minks, when he was in the lab, chose to look at the A repeats. If they were critical for silencing, could they alone account for the silencing? And so in the same model system, he integrated only a kilobase of XIST containing those 5' A repeats, and again looked at silencing of the adjacent reporter. And you can certainly see that the A repeats alone are quite capable of silencing. So Jakob went on to generate a consensus loop that he called, which was one copy of the A repeat. In the endogenous XIST, there are 9 copies of A, so he assembled 9 copies of the loops, and sure enough, that could silence fine, suggesting that the variability within the A repeats was not contributing to its function, but also allowed him to then aggregate together different numbers of loops. And what he saw was that additively, from 2 repeats, you could get more silencing the greater number of repeats that you had present. However, when he looked beyond proximal reporter constructs, he could not see silencing of endogenous genes. So he established a collaboration with the Lefebvre lab and Danny Leung and other members of the Brown lab, including Angela Kelsey, Christine Yang, and Thomas Dixon McDougall, to look at the full length XIST and how it was functioning. And what they compared was 9 different integration sites of XIST. So these were the same construct flipped into 9 different FRT sites. And in each chromosome, and in fact, in any chromosome we've examined, XIST is able to localize. And as a quick measure for silencing, you can look at COT1 hybridization to the RNA and see that where XIST is, you see a depletion of repetitive elements from that chromosomal region. This work has been published, so I'm just going to summarize a number of these results. So first off, I'd like to emphasize that human and mouse differ in their XIST sequence and repeat structure. So we want to study human XIST. In addition, as Brad has told you, human and mouse differ in the number of escape genes, and there are also differences in regulation of XIST. So we've established a model system using an inducible XIST in somatic cells. The A repeats are required for silencing, but are sufficient only for local silencing. So we need to understand what the additional domains of XIST are doing. XIST can localize to any chromosome, but the integration site controls both the extent of endogenous gene silencing and which chromosome features can be established on that chromosome. Despite this variability, from those studies, we could not establish a clear hierarchy of factors. So for subsequent experiments, we're going to focus on the 8p integration site, which recruits a variety of chromatin modifications as well as silencing. Interestingly, the recruitment of K27 trimethylation seems to occur at pre-existing locations on the chromosome. So the experiments I'm going to tell you about that have not yet been published are from Thomas Dixon McDougall, and this is his thesis work. He wanted to dissect XIST function by creating deletions along the length of XIST. So in addition to integrating constructs with different deletions, he used CRISPR to delete specific regions of repeats or non-repetitive regions across the XIST RNA. And then used FISH combined with immunohistochemistry to study the accumulation of marks at the XIST focus. So as you can see here, the full XIST and each deletion was still able to amass XIST in a focus in the nucleus, and this tended to be at the nucleolar periphery. The only exceptions to the ability to coalesce XIST were a deletion of this region and also this large double deletion region where you can really start to see that XIST drifts through the cells. So then Thomas looked at the recruitment of various chromatin modifications or other proteins to this focus of XIST. And for each construct then and each mark, he looked at over 60 cells. And what I'm going to show you are the aggregate Z-score charts. So the deletions correspond to these colors, and you can see that for H2A ubiquitination, when we delete the A repeats, we are no longer able to recruit ubiquitination. Surprisingly, there actually appeared to be 4 different regions of XIST that were required for this recruitment. Now, this region of the BC repeats was not surprising. That's the region that has been reported in mouse. It's been reported that that region binds HNR and PK, recruits PRC1, which leads to ubiquitination of H2A, which then enables recruitment of PRC2 and leads to H3K27 trimethylation. So an obvious question is, are these same domains required for recruitment of that mark? And the answer quite dramatically is no. It's very different domains that are required. So here, well, the A repeat was required for ubiquitination. It had only minimal effect on K27 trimethylation. Well, the E repeat region was required for the K27 recruitment of ubiquitination with that region was fine. So very different regions are required. And that's different from the story in mouse in embryonic stem cells. In mouse, the ubiquitination of H2A has also been shown to be critical for the recruitment of SMUSH-D1, and which is involved in subsequent DNA methylation of a proportion of the genes on the X. And so Thomas looked at SMUSH-D1, and sure enough, some of the regions are conserved, including this region that's the same as is seen in mouse. But additionally, a region of non-repeats, 3' to the D repeat, is required for SMUSH-D1 that was not required for ubiquitin H2A recruitment. Another common feature examined for the inactive X chromosome is macro H2A. And so Thomas did the same analysis. And the pattern that he sees there is somewhat reminiscent of that seen for H3K27 trimethylation. So I've loaded that image again so that you can see that there is overlap, but also regions required uniquely for each feature. So to validate that PRC1 and PRC2 may be independent of each other in these human somatic cells and upstream of SMUSH-D1 and macro H2A, Thomas went on to use inhibitors. And so by inhibiting PRC2 with GSK 343, he saw loss of trimethylation on the inactive X. There was approximately 75% overall drop in H3K27 in these cells. And that also led to loss of macro H2A recruitment, but had no significant impact on ubiquitin H2A recruitment. And vice versa, PRC1 inhibition, in this case only impacting about 50% loss genome-wide, led to almost complete loss of ubiquitin H2A on the chromosome 8, but no impact on K27, but yes, eliminated the ability to recruit SMUSH-D1. So just to summarize, this is what I've said, that it appears that PRC1 and PRC2 are independently recruited to different domains and are upstream of SMUSH-D1 and macro H2A. Strikingly, for all of the factors that Thomas has looked at, there are multiple domains required for recruitment, with regions in between those domains which can be removed and not impact recruitment. One explanation for this is that there is contact across those domains, and in fact, Howard Chang's group has shown such contact. Multiple groups using Paris have shown that there are interacting domains, and the Chang lab report that there are regions that accumulate different classes of protein, and of note, using PERCH-seq, they see long arcs for EZH2 recruitment, suggesting that yes, there would be domains that are required with other domains in between that would not be required. So I'd also like to discuss the impact of this on silencing and slide in a few additional marks here. So for silencing, when we did RNA-seq, Christine Yang identified genes with expressed alternative alleles, and that allows us to examine silencing by allelic expression. So most of these start with approximately 50% expression of each allele, but within 2 days of DOX treatment to induce XIST expression, you can see that we achieve approximately 80% silencing of these genes. So that then allows Thomas to go back and look at all of his deletions again. And in the next slide, he looks at normalized silencing. So if full XIST allelic silencing is 1, we can see that deletions of the A repeat region extending actually up to the F region completely aggregate silencing, but also that a region at the 3' end of XIST is necessary for silencing. So what can be responsible for this? Going back to the Chang lab work, we can see that the A repeat, as well as the other repeat regions along XIST, as well as the non-repetitive region of exon 6, recruit a variety of proteins. In fact, we know several hundred proteins who seem to enrich with the inactive X. But one of these stands out. If any of you saw Howard Chang's talk earlier this week, SPEN is recruited by the A repeats and is believed to then recruit HDAC3 and lead to deacetylation. So why do we also see a requirement for the 3'? And we wonder if that would not be because of RBM15, who's responsible for adding M6A modification to XIST, not only near the A repeat and the F enhancer region, but also at the 3' end, which then recruits YTHDC1, which has been shown in mouse to be also responsible for silencing. So Christine Yang in the lab has gone on to do cross-linked immunoprecipitation to start identifying which proteins interact where, and does indeed see that SPEN will interact with those 5' A repeats that Jakob had originally shown could do proximal gene silencing. Okay, so I'm up to my 20 minutes. I will just mention that the E repeat also is known to bind a number of proteins, and these are believed to be actually involved in multivalent interactions with proteins that have these intrinsically disordered domains. and therefore function cooperatively to seed condensate, which may contribute to the inactive X. So this is what I've told you. There's a lot of crosstalk between the factors that are recruited by XIST, but there are also pathways where we think that PRC1 is functioning before recruitment of SMUSH-D1, for example. So I'd like to thank the members of the lab who keep helping try and solve the puzzle of XIST, but in particular acknowledge Thomas, Dixon McDougall, whose work I've presented. And I'll stop there and take any questions.
Awesome. Thank you so much, Carolyn. You really define this field, and it's amazing to what depth you guys have been able to characterize the process of X inactivation. It's really fantastic work. I have a few questions here. So a natural one for me would be to ask you if macro H2A1 or macro H2A2 seems to be more involved in what you're observing, right? I'm assuming it's A1, but.
We have actually not differentiated them, and our antibody will recognize both. And both have been seen to recruit to the inactive X in mouse, but we haven't distinguished.
Okay.
Do you think we should?
And I don't know if it's super important, but it's yeah, I'm just a curiosity, right? Because at least we are seeing that the two variants of macro H2A are doing such different things, and Emily Bernstein, I think, reported very different functions for the two isoforms. So not isoforms, sorry, for the two different variants. So it's always a curiosity of ours, right, to look into that kind of stuff.
The isoforms, but I think an additional important thing is that it's been shown to be modified on the inactive X, so that's also important.
Makes sense. Ben Laufer has a question. He's asking, do the genes that escape X inactivation show enrichment for any gene ontologies or pathways?
Not that we have ever been able to discern. As Brad briefly mentioned, there are reports that XIST tends to fall off some of the pro-B cells, and that seems to allow expression or reactivation of some genes, perhaps accounting for some of the tissue specificity, in which case you might say immune, because that's where it's been seen to fall off. But I don't think it's so much a gene ontology as an XIST stability issue.
Yeah, makes sense. And then I'm kind of curious about the different isoforms of XIST that you alluded to in the first few slides. So do they have actually different functions? Do they have different localization patterns along the chromosome or interaction partners in terms of RNA protein complexes?
We really haven't seen any. We are quite focused, sorry, way back, on the short isoform just because it was so much more clonable. Most of the protein interactions are focused in A repeat, this broad region, E repeats, and actually the terminal bit who comes back to be on the short isoform. But if that really didn't matter, why would you keep it? The other odd thing is that there's described a P2 that you can actually start transcription at approximately the F repeat, which again makes no sense because the A repeat is the critical part for silencing. So would those actually do anything? It seems unlikely, and we think more likely they're just an outcome of recruiting transcription factors to an enhancer. So we haven't studied them.